Frank Tagliaferri is up first, and he on an exenatide transdermal patch program that I think he's going to tell you about. Thanks, Rick.
Okay, so I'd like to thank the organizers for the invitation to come speak to you today. Like I said, Chris said, this talk's going to be kind of a little change up from what you heard this morning. I'm going to really try and focus on kind of a review of transdermal technologies, kind of where they've come from in the last 30 years. I'm going to try and focus on how these new technologies hopefully can open up some avenues so we can start delivering molecules like the peptides everyone here is working on through the skin, which I think is a pretty valuable alternative to delivering molecules with just injections. Just some kind of background on the transdermal delivery market, just to kind of show you that it does it exists, but it is a sort of modest form. The market, I guess in 2010, was kind of listed at about $21.5 billion. And pretty much all that market was really due to first-generation patch technologies. So these are kind of the traditional passive transdermal patches that people know, nicotine, fentanyl, nitroglycerin, estradiol, et cetera. And that technology, and I'll go over a little later, is really just limited to small lipid-soluble molecules, of which most of the new molecules coming out nowadays don't really fit into this. Even so, within the next 5 years from 2010, it's expected that the market for transdermal patches is going to grow by about another $10 billion. And that's really due to advances in transdermal technologies that will kind of drive the market growth, but it's also going to kind of open the opportunities to deliver molecules through patches. And so you'll have more products that are available to the technology, and hopefully that'll turn into more products for patients. Again, just why do you want to look at transdermal patches? Again, just some Frost and Sullivan market research is transdermal patches really have a high acceptance for patients. They like them. They're easy to put on. They're kind of non-invasive, and the compliance to putting on a patch is really second only to oral drug delivery. So each day get up and put a patch on. That's almost as easy as kind of taking a pill, and that's kind of what most of the people interviewed were talking about. Physicians also like patches because, again, of compliance. It's much easier for a physician to send the patient home with a patch and have some confidence that they're going to put that on every day, as opposed to sending them home with an injection that they have to rehydrate and inject, et cetera. So I think physicians prefer patches also, just because the adherence will be better and the outcome of therapies presumably would be better. This is just, again, some more research in terms of where they asked diabetic patients. They gave them the choice of any delivery technologies you can think of, and asked them, what would you rather use? And kind of not surprisingly, the thing that was most popular was oral delivery. So people, it's easiest to take a pill. But over here, kind of the second most popular form of delivery was really transdermal or topical, and it kind of had the highest acceptance factor and kind of fairly low kind of unwillingness to use factor. And again, not surprisingly, injections were probably the least popular form of delivering a therapy for diabetes, with fairly high kind of unwillingness factor. I think what's not really explicit in this slide is the fact that those patients that say they don't really want to use injections are not really saying it simply because they're afraid of needles or they're afraid of the pain or the inconvenience of giving injections. If you look at the research with diabetic patients, it turns out that a lot of them don't want to do injections because it sort of makes them come to the realization that they have kind of a chronic degenerative disease that they're going to have to treat for the rest of their lives with an invasive injection. And so they'd really rather procrastinate and kind of put off the injections as late as possible, so they don't have to really accept the fact that they have a disease. And that's really sort of unfortunate because, like you heard this morning, something like GLP-1 has a lot of benefits, and if people could start it earlier, they'll probably help the progression of the disease and their outcome. So I think anything that gets these people on these therapies faster is really going to help their outcomes and improve their quality of life. So if we think about, I'm calling it transdermal drug delivery space, but it's really all drug delivery space. If we think about where current transdermals are in this space, it's down here in the lower left corner. And like I said, these are highly lipid-soluble drugs and small molecular weight, several hundred Daltons. As you get up in water solubility, you can start to access some of the orals. But as you start moving into larger molecular weight and higher water solubility, you get to all these kind of drugs out there that everybody's working on now that can really only be delivered by injection. And so the current kind of passive market is very small, but if we can get some of these transdermal technologies to kind of open the space, you can see that the opportunity for turning all these molecules into patches is there. I couldn't give a transdermal talk without a piece of skin. So just to kind of orient you, what we're talking about here is the kind of hardest barrier to transdermal delivery is really the smallest part on the skin here. It's this thin layer of stratum corneum on the top of the skin. It's highly hydrophobic, it's really dead cells, and it's really impenetrable to anything that's hydrophilic. There are some access points to the stratum corneum, hair follicles, and ducts to sweat and sebaceous glands. But the real key is to actually get stuff past that. Once you get it into the epidermis, then you can rely on things like diffusion to traverse drugs into the circulation and start to pick up by blood vessels. Just again to show you where transdermal technology started was really passive patches. There's all kinds of varieties out there now. There's reservoir patches, which really just basically a drug solution that sits on top of the skin. There's matrix patches where it's in some kind of polymer matrix. Drug and adhesive where you can dissolve a hydrophobic drug in something like a silicone adhesive. But again, it's low molecular weight, lipid soluble. The mechanism is fairly straightforward. They're simply absorbed into the hydrophobic stratum corneum. They really form a depot in there, and then eventually diffusion passes it from the stratum corneum into the epidermis and the dermis, and eventually into circulation. As I talked about, most products to date are small, even though they do make lots of money. Fentanyl patch is probably one of the biggest out there, but nicotine, lidocaine, estradiol, et cetera. Rotigotine is probably one of the newer transdermal patches. People have tried to add novel enhancements to this technology to kind of open the space, but to be honest, none of these enhancements are really opening it to the kind of molecules that we're probably interested. And so there's penetration enhancers, vitamin E phosphate, even some peptides. People are adding, Prausnitz's group at Georgia Tech added Meganin peptide to hopefully cause pores, but I'd say the results were fairly modest. People are also using things like heat-assisted delivery to help increase the permeability of skin, but it's not really increasing the permeability enough to get hydrophilic molecules across. So I'm going to kind of go through some of the technologies where people have actually had some success delivering some larger biomolecules. The first is something people are probably familiar with. It's been around probably for close to 30 years, and that's ionophoresis. Ionophoresis really is the application of an electrical current to a drug that's placed on the skin, which causes the drug to pass through the stratum corneum and into the skin. I sort of have just kind of one example of how an ionophoretic process would work here, in which you have a positively charged drug on the surface of the skin and a positive electrode. You put current in there, and you basically get an electrorepulsion, where the positively charged drug is repulsed from the anode and basically enters into the stratum corneum. There's another mechanism that goes on here also, an electroosmotic mechanism, which takes advantage of the fact that the skin is negatively charged. And if you put a drug on top of the skin with a positively charged solution, you can sort of get a bulk flow of the solution through the skin, and that kind of carries with it any dissolved positively charged drugs that are there. The magnitude of permeation is really proportional to how much charge you're putting on the skin and the amount and the duration of the current that you're applying to the skin. It's also proportional to the size of the electrode. If you had the ability to wear a giant electrode, you could deliver quite a lot of drug, but the way these systems are set up right now, you're probably limited to very modest amounts of drug, I'd say in kind of the microgram to maybe milligram range. The thing to keep in mind with this technology is, even though it is technically transdermal, the transport's mainly through hair follicles, sweat glands, and sebaceous glands. So you're not really creating any permeation in the stratum corneum. You're really relying on the fact that these charged molecules kind of track down the hair follicle, eventually get under the stratum corneum, and then form a depot. And that depot eventually diffuses into the circulation. Again, just some advantages from the ionophoretic method. First of all, it's transdermal delivery of small and medium-sized charged molecules. So that's the other thing. You're going to need a charge to deliver these with the current. The rest of the advantages really have to do with almost all transdermal deliveries. I should have talked about it earlier, but they all do avoid hepatic first-pass effects. So You don't have to worry about that. They also avoid the gastrointestinal degradation, which is obviously a problem, especially for things like peptides. For the most part, they can all be user applied. And the other thing, which is maybe a little subtler, more subtle anyway, is that you can get a rapid dose cessation once you remove a patch. So as opposed to some of those passive patches where it may take 20 hours to get a steady state because you're forming a hydrophobic depot in your stratum corneum, with these active methods, you're only getting delivery while the current is applied. So if you take the patch off or pull out the battery or whatever, the drug will stop. And so if you had any kind of adverse event, you could stop delivery immediately, which is really counter to even some of the things we talked about this morning, where you're putting a month payload on a person that they can't ever remove from their body. So some of these transdermal methods really have that advantage. Some of the disadvantages, drug needs to be in ionized form, may not be as important for something like a peptide, but for some small molecules, it is an issue. You do have a pH gradient as you're delivering drugs through the skin with anapheresis. Top of the skin is probably around pH 8, and it's getting lower as it goes through. So you go through a lot of trouble to get your drug at the pH and the charge that you want, but the body really kind of fights against that and could change your charge. Probably the most important drawback is current actually causes pain if it's not done properly. You can get skin irritation, erythema. Worst case, you can get burning and blistering. There's also some practical limits to the electrode size, like I talked about, how big a patch you can wear, and the size of the battery. I do think, however, that this is the one area where anapheresis may see a little boom, is just the advances made in battery technology. You can make smaller and smaller batteries which hold more and more current, and they can be shaped in any way you really want. So I think that'll kind of be one benefit, so you don't have like a D cell hanging off your arm while you're wearing these. But again, the typical conditions, you put a couple milliamps on your skin for 20 to 40 minutes to deliver the drug. So you kind of have a limited window for how long you can deliver this and how long the body can actually tolerate the current. Just some formulation considerations. As most formulations, it's really dependent on the concentration of the drug, the charge of the drug, the size, the pH. The thing you have to be really careful about is ion competition, because as you're pushing positive ions one way, you have to rely on negative ions going the other way to kind of complete the circuit. Buffer compatibility then becomes an issue. You have to make sure your peptide or your drug is going to be compatible in this electric buffer that you're using. And then this is almost always delivered as a solution, so you're going to need solution stability, whereas some of the other technologies I'll talk about, you can use a solid formulation. There's been quite a number of systems that are commercialized, but I'd be hard-pressed to say that any of them are extremely successful. Most of them are small molecules. Probably the newest one is the liver of sumatriptan using anapheresis. Vitaris had a lidocaine system out there. J&J had their ANSYS system with fentanyl hydrochloride that was on the market for a while, then pulled. And then some people sell things basically which are empty patches with ionophoretic electrodes on them that they can use for whatever they want to put in them. People most of the time use things like glucocorticoid steroids or lidocaine for physical therapy. Like I said, these molecules have been, this system has been used for peptides, mainly because some of them are fairly potent and you can deliver just micrograms in their charge. So things like FSH, LHRH, insulin have all been delivered. And then, like I said, some small molecules have gone through, but I think it's really hasn't really seen the boom that it probably could have. If you look in the literature, there's probably more articles from academic labs on anapheresis than any other transdermal technology. Another active technology I'll touch on is sonophoresis. This is the use of sound or ultrasound to basically increase skin permeability and drug permeation and get drugs past the stratum corneum into the skin. There's sort of two methods people employ to carry this out. There's high-frequency sonoporation, so that's up to about 16 megahertz. I think the increase in delivery is sort of modest. You get about a 1 to 10-fold increase. The mechanism here is you really get cavitation of droplets, et cetera, within the skin or the skin appendages, such as the hair follicle, that actually helps the permeation and gets drug in. To date, most of the high-frequency sonoporation has been really limited to small molecules. A little more recently is low-frequency sonoporation, really because these devices just became a little more available on the market. So this is about 20 to 100, maybe up to, say, 700 hertz sound waves. I've seen some fairly impressive increases in delivery. You can get 100 to 1,000-fold increase in delivery. Mechanism's a little different here. You're getting acoustic cavitation above the skin. So basically, you place your drug or your coupling solution above the skin, and you'll get cavitation in there, which almost forms a microjet, which kind of almost punches a defect in the skin, which allows molecules to go through. And like I said, this technology has really been shown to work pretty well with larger molecules, though it is fairly new. But things like chymotrypsin, amphotericin B, inulin as a model compound, and something as big as EPO antigens and nanoparticles have all been delivered through the skin with sonophoresis. Again, there's not many commercial sonoporation devices out there. This one was out there for a while. I actually don't think it is anymore. It's just a sonoprep device where you kind of pick your drug of choice and then use it on the skin. I included this reference that I just ran across a couple weeks ago. This is from Langer's group at MIT, where he kind of combined the high frequency and low frequency sonoporation ideas. So you simultaneously hit the skin with both. And he showed tremendous advancements in the delivery of high amounts of large molecules. So we're talking now, instead of just micrograms, he was able to get milligrams of something like inulin as a 7,000 Dalton molecule through the skin. So I just kind of included this to show that maybe this technology can see a little resurgence, because the devices themselves have gotten better and easier to use, and by combining a couple of technologies, you're able to deliver some larger molecules. Next part I'll probably talk a little more in depth about, because this is a skin ablation technology. This is kind of a field I've been in for about the past 8 or 9 years. The technology I'm going to talk about is microporation. So microporation really uses energy to create small micropores in the skin, and it does this by ablation of little parts of the stratum corneum. So as you remove pieces of the stratum corneum, you actually expose the under side of the skin, and you get a hydrophilic channel, which allows both water-soluble and large molecules to pass through. It's done by a number of techniques. People use electrical energy, radio frequency, or laser energy to create these holes in the skin. As long as it's done properly, it's fast and it's painless, on the order of a few milliseconds. And if it's superficial enough, you're not the energy is not deep enough to contact nerve receptors, so you actually don't feel any pain. Thing to keep in mind, it's actually a passive delivery once you do the application. So as opposed to anaphoresis, where you have to keep the current on the skin while you wanted the drug to deliver, microporation, you really treat the skin with the device, create the holes, remove the device, and then basically put a passive patch on top of the skin, and you rely on diffusion to deliver the drug from the patch into the body. I'll show you a lot of examples, but you can deliver both large water-soluble molecules as well as small water-soluble molecules. It doesn't work as well with lipid-soluble molecules, because you've kind of removed stratum corneum, and you now have an aqueous environment that your drug is going to sit in. Just several examples of the kind of molecules that have been delivered. Peptides like PTH, exenatide, calcitonin, larger molecules like insulin, growth hormone, GCSF, and then we've gone up to things like antigens and antibodies, and even plasmids have been delivered this way. And like I said, you can deliver small molecules, but they need to be water-soluble. So as opposed to duragesic, which is fentanyl base, you can deliver fentanyl citrate, the water-soluble salt, in this way, things like lidocaine, hydromorphone, et cetera. The other great benefit of this technology is, depending on what size patch you want to use, you can deliver very high doses of drug. On the order of a couple hundred milligrams of small molecules have been delivered this way. And then I'll show you later with some formulation methodology that you can now get sustained or bolus profiles. It's really just a matter of how you make up these passive patches to deliver drugs. So some of the companies that kind of pioneered this, Altea Therapeutics, where I was in Atlanta, Georgia, it uses thermal ablation, and this was done with basically a small battery where you heat up a disposable metallic array. It's a common theme you're going to see here. The Altea's assets were sold to Nito Denko last November, so it's no longer around. But before then, like Chris mentioned, we had a large project with Lilly and Amylin to deliver GLP-1 in a transdermal patch. We also delivered a number of peptides from Kai this way. We had a project with Hospira to deliver enoxaparin sodium, and we had clinical programs on insulin and interferon alpha, as well as a number of other molecules. Another company involved in this space was Transpharma Medical out of Israel. They had their Viador system. They use radio frequency ablation, so radio frequency on a small electrode creates holes in the skin, so they had a disposable microelectrode array. Again, common theme. They had a project with Eli Lilly to deliver PTH 1 through 34 in phase 2. I think that didn't meet its endpoint, and at the end of last year, and the assets were sold to Syneron in March of 2012. But again, they delivered things like PTH, GLP-1, calcitonin, insulin, and growth hormone. Final company on here is Pantech Biosolutions. They use a laser to create the holes in the skin using a disposable interface. I do think they're still around in Europe, and they've delivered a couple molecules, triptorelin and FSH. Just a little more detail in terms of the mechanism here. This is just showing you kind of a cartoon of skin with the hydrophobic stratum corneum on top. A hydrophilic molecule really is just going to sit on top or bounce off. It's not going to penetrate. However, once you use microporation to create holes in the stratum corneum, you now create these aqueous channels which have access to the viable epidermis, so the drugs can penetrate and then diffuse into the dermis and then get picked up by the blood vessels to deliver. Just to show you a little visual what's going on here. This is just what a metallic array looks like in the Altea system. It's a copper-clad thin piece of foil in which some of the copper has been etched away to expose little stainless steel filaments. There are areas of high resistance, so when you put a current across this film, those areas heat up. You do it quick enough and with enough energy, you create little, when it's against the skin, you'll do little flash vaporization of little holes in the stratum corneum. This is sort of what it looks like if you were going to put that device on a rat, do the microporation, and then just take the device off. And here I just stained it with green food coloring just to show you the little holes that are created in the skin. This is 120 holes in a 1 square centimeter area, and it was done in just 2 milliseconds and then removed. You can get a little fancier and kind of use something like a fluorescent dye. This is calcine, where you can actually measure things like the intensity of the dye, the penetration, and do things like measuring the size of the pores, etc. If we want to look at some histology of what happens, this is basically some pig skin where you're looking along an array of filaments here. So you see at very regular intervals, you get little areas where you've removed little pieces of stratum corneum. And if we just kind of zoom in on one, this is sort of what it looks like. That kind of dead, feathery stratum corneum has really been kind of blown away, and you're just starting to impinge on the viable epidermis. The size of these pores, they're probably about 40 to 60 microns deep, and depending on the size of the filaments, probably about 50 microns wide. So they're certainly big enough to deliver any size particle through these. Just to show you the kind of mechanistically how it works, I realize this is a small molecule, but it kind of shows the differences between some of these ablation or active technologies versus how they would work with a passive system. The curve on the bottom, this is a hairless rat study where we put a Duragesic patch on a rat and just measured plasma fentanyl levels. So Duragesic has fentanyl base, the hydrophobic version of fentanyl. As it stays on the animal's skin, it takes quite a long time before it gets to a Cmax. And then if you take the patch off at 24 hours, it then takes, again, almost another 20 hours to decay to baseline because you're forming this depot in the skin, and then it eventually has to leave the depot and diffuse into the circulation. If, however, you do the same sort of study, but at time 0, you microporate the animal, so in a 1 square centimeter area, you make holes, and now you put a patch on the animal that contains fentanyl this time it's fentanyl citrate. The fentanyl citrate's in a solid form now. It's just, we suspend it in a polymeric matrix and just dry it down, so it just looks like a thin film of drug. If you put that over the holes are basically hydrophilic, and interstitial fluid comes up and wets the film and dissolves the drug. So then the drug basically get a fairly rapid rise to steady state. It stays there as the drug is delivering for 24 hours. Then when you take it off, you get a more rapid decrease. Just maybe specifically for this audience, I wanted to give you just an idea of some of the peptides and the small proteins we delivered with this system, just to kind of show you that it's not really dependent on anything, molecular weight, charge, et cetera. So some of the peptides, PTH 1 through 34, we did deliver it in a bolus profile, and we got preclinical bioavailabilities anywhere from 80 to 100%. A couple undisclosed peptides that we worked with that are fairly highly positively charged. We developed both bolus and sustained delivery systems for those. Like I said, GLP-1, I'll show you in a second, calcitonin. Then we get into small proteins like GCSF and a molecule like insulin we could deliver either as bolus or a sustained delivery. This is some human clinical data where we delivered insulin to healthy subjects. In this study, the subject was brought in, we microporated, put an insulin patch on them, measured the serum levels for 24 hours, and then the patch was removed. So this is just insulin pharmacokinetics. As the patch is on, you get a fairly steady state. You take the patch off, and it drops to baseline. In order to show that it is bioactive, we just also hooked the subjects up to a glucose infusion. So as the insulin was delivering, in order to keep homeostatic levels, as insulin's delivering, we had to turn up the glucose infusion pump. And so while insulin's delivering steady, we had to keep the glucose pumping in steady to deplete the insulin. And then when you take the patch off, there's a slight delay, but then you could start turning down the glucose pump in order to keep them at a steady level. And so this is a 12-hour delivery. We've also did it in 24 hours, and we've done it in both normal subjects and type 1 and type 2 diabetics. So to kind of follow on to the theme this morning, we did have a fairly large project looking at delivering GLP-1, specifically exenatide, in a transdermal patch. I'm going to show you just some preclinical data here, but we had some clinical data also. What I'm showing here is two different formulations we made of GLP-1. They're both solid formulations where we take a lyophilized powder, we suspend it in a organic solution along with a dissolved polymer and some excipients, and then just dry that down to a dry film. And so the film contains exenatide suspended in a polymeric matrix. This is just some dissolution data. So this is what we consider our sustained delivery formulation. If you take that film and just drop it in a dissolution apparatus and measure exenatide released over 24 hours, you see you get a fairly steady rate over 24 hours. The other formulation we were interested in was something maybe that mimicked more of a Byetta sub-Q injection. So this is a more rapidly releasing formulation where we really minimize the amount of polymer holding the drug in formulation. And so you get kind of a rapid release of drug. And so these films were placed on, this is a hairless rat study where 1 square centimeter area was microporated. And so you could see, in this case, the extended formulation. You microporate at time 0 and put the patch on. You could rise to sort of a steady state, and then when the patch is removed, it falls to baseline. Alternatively, if you want a more rapid formulation, this time you put it on the skin, and the drug really immediately dissolves from the film and enters the circulation. I don't have the data to show today, but we've actually tested kind of both these formulations as well as a couple others in both normal subjects and people with diabetes, and we showed kind of expected pharmacokinetics as well as glucose lowering. So I think it does have some promise to deliver GLP-1 with microporation in a fairly small. These were all 1 square centimeter, even for the humans, 1 square centimeter transdermal patch. Last thing I'm going to touch on is microneedles. I'm not going to go into this into a lot of depth because Peter will talk about his project in a moment. But just to give you kind of an idea about how microneedles work in terms of delivering drugs through the skin, they're basically minimally invasive needles, typically less than 1 millimeter in length, that use a kind of a mechanical perforation of the stratum corneum to deliver molecules. I'm going to say large molecules, but they'll really deliver almost any kind of molecule you're interested in. There's all kinds of flavors of microneedles. They could be solid, hollow. You can coat microneedles. You can actually make the needle itself out of the drug, et cetera. You can make them out of a dissolvable substance like a sugar, et cetera. For the most part, a lot of the data is on bolus administration, but there's some sustained delivery out there also. There's a number of kind of players here. Some of them are around, some of them aren't. But people have really been working quite a while, and I think microneedles are probably you'll start to see more and more commercialized over the next several years. Just the one company I'm going to kind of focus on here. At 4P Therapeutics, we have a collaboration with Kimberly-Clark, who has a new microneedle technology. They call it their biologic transdermal system. along with the fact that they have a microneedle shape that can go through the skin and deliver, they also have a proprietary coating that they place on the surface of the needles. It's a nano structure that's been specifically designed to modify skin permeability. So in vitro and in vivo studies, it's been shown to modify cell permeability, in some cases cell membrane shape, and also can elicit different secretions of cytokines. So their thinking here is by placing this on microneedles, you're not only relying on the hole you're creating in the skin to deliver the drug, but you're relying on the nanotechnology structure on the surface of the needles to affect the permeability of the cells and allow more molecules or more amounts of molecules to be delivered. So they're working on both a microneedle structure, but they also have some implantable drug delivery systems that they're working on. This is just one quick early study we did with them treating rabbits. This is using entanercept or Emerald. So you got to remember, now we're talking about a molecule that weighs 150,000 Dalton. So it's a very large molecule, and with microneedles, you get fairly modest levels of Emerald delivered through the skin with these microneedle systems. However, so these are naked so-called microneedles, but if you cover the needles with the nanostructure, you can now increase by several times the amount of delivery you're getting in the skin. So again, this is fairly early. There's some newer data coming out, but I think it shows promise that you're basically taking a technology that's might have been around a while, and you're trying to improve on it and able to deliver more molecules and larger molecules. So I'll just kind of try and summarize here. I hope I showed that traditional passive transdermal patches kind of have limitations. There's not really that many candidates left, or there's just not coming. Anything that could be tried in a transdermal patch really has been tried, so we're going to have to wait for new molecules to be invented. There's obviously need to deliver biologics and water-soluble drugs in methods other than injections. I think every new biologic out there is always a sub-Q or an IV infusion, and I just think you can get a much better uptake in the population and really help people that don't want needles or are afraid of needles if you can get these systems delivered in another more user-friendly form. There's a number of technologies in development. I think the stratum corneum disruption, kind of microneedle, microporation, probably has the best possibility to deliver these molecules, because you're actually breaking the stratum corneum and creating holes which these molecules can fit through. Some of the active systems, you really got to rely on an outside power source to do it, but they can deliver modest amounts for drugs that you don't need as much delivered. I think commercialization of any of these new technologies is going to kind of expand the utility of transdermal delivery. I think there's a much bigger pool out there than transdermal can touch right now. And like a lot of people said this morning, most of the new drugs are these biologics, and so we really need new ways to deliver them. And the other thing I think is important, you saw that there was something like 40 approved peptides right now. They're probably all injectables, but if we can get this transdermal technologies to work, it provides a lot of follow-on opportunities. So you release a drug as a sub-Q, but then follow on with a transdermal. And then kind of what I think is the holy grail, and we're working a little with some companies on this, is basically the potential for the first dosage form to be commercialized to not be a sub-Q injection. So have a molecule that maybe not designed from scratch, but that's been chosen to go into the clinic because it matches the qualities that are necessary to deliver transdermally. So just acknowledgments, Altea Therapeutics, Kimberly Clark, and some of the data I showed from Eli Lilly and Amylin. So thanks for your time.
Time for questions, if anyone has any. I have a quick question, Frank. On the etanercept example, the microneedle was administered and removed after 10 minutes or left on for that whole duration?
These are microneedles with a channel, so it's a liquid reservoir, so the fluid can travel down the reservoir. So these were simple liquid reservoir patches where the drug remained on for the 24 hours.
Okay.
They're dissolvable, right?
This one particularly was not dissolvable.
But there is needed dissolvable.
Oh, sure.
The 3M company is working on that, right?
Yeah. 3M, it's a little confusing, some of the space. A lot of people that claim they're using microneedles are really using intradermal needles. Right. So I always like to look at the length, but I've seen some Humira data where the needles were about 1 millimeter or 1.5 millimeters long. So I'd call that more intradermal. But yeah, people make lots of dissolvable needles.
And the payload, how much you can put? It depends on the
For a dissolvable needle, I think you're a little less. Maybe Dr. Dodana is better to ask, but I'm guessing a few milligrams is probably your maximum that you can put on a dissolvable needle. If you can get it to deliver as a solution, you're sort of just limited by the concentration you can place in your reservoir.
So it depends. And I have also market research slide long time ago. When they asked the patient, what do you want? They told us, they keep saying the patient wants a safe and drug without any side effect. So the way of administration will come number 3. So between oral and transdermal is really later on. I mean,
No, I agree. But that's why something like exenatide is kind of an issue, because you have to deliver with an injection, and it may make you nauseous, and you may have to do it twice a day. So there's issues with some of those.
But I still think the transdermal is the way of the future. At least I love it. I don't know who else loves it.
So I have more of a general question. If you apply the transdermal at various places, arms, legs, and so forth, does that have an impact on uptake and distribution?
It sort of depends which technology you're using. So something like passive delivery has a definite issue. People usually indicate for the arms, so they get constant delivery. You don't want to put it on something that has thick skin, like the sole of your feet or something, or thin skin, like the back of your neck. So that has an issue. Some of these other technologies where you're actually making holes in the skin, it's not necessarily as critical, because you're actually puncturing a hole in the stratum corneum, so you don't have to rely on...
And more generally speaking, does a subcutaneous or skin delivery like this get the same distribution as intravenous?
The few studies we've done, we've seen very similar distribution, because in essence, you're almost delivering from the same sort of space as a subcutaneous depot. And we found sort of the similar immunogenicity also, but I guess it's going to depend on the molecule.
If you have a chronic indication and the patient has to put these patches, let's say I'm talking about microporation or microneedles, day after day after day, what will his skin look like? And is there any potential for infections? If you have that many holes?
It's really, they're almost always indicated to rotate the sites. Even passive transdermal patches, you're supposed to not use the same site except once every 7 days or something like that. So that's what we would do. I mean, the kind of goal when you're developing these systems is to have the cosmetics good enough so you actually don't see the site after you take a patch off. But we wouldn't indicate for you to, say, microporate the same site every day. Simply maybe move. We used to indicate for 2 arms and 2 legs and then kind of rotate.
Yeah, but even if you move it, do you see marks from your previous applications?
It really depends. I mean, some of the systems we've used, the marks disappear within, say, 24 hours. I admit when we were first developing this technology, it wasn't the case. But I think if you lower the energy so the pain is tolerable and the cosmetics are right, you can optimize kind of how much residual irritation you see. I mean, but the truth is, you look at people that get, daily sub-Q injections. A lot of times they'll have, welts and irritation there also. So it's also drug dependent.
You did not say the bioavailability. I keep hearing between 60 and 70%. Is that true?
It really depends on what you're delivering. When we're delivering small amounts of molecules, a lot of times, like I said, we can get high bioavailability. Other times, the bioavailability is going to go down. It's like insulin. We did a number of clinical studies. We can get up to about 24% bioavailability delivering insulin in a patch.
Not the 60, 70% we hear?
With insulin, no. With something like...
GLP, how much?
I don't really want to go into that.
So less than 20?
It could be.
The insulin data was humans, right?
Yeah, insulin was human data. And, we've delivered molecules like fondaparinux and got, 90% bioavailability, and heparin or enoxaparin and got, say, 30 to 50% bioavailability. So it sort of depends on what molecules you're delivering.
Because in 3M, they claim 65 to 70%.
Yeah, like I said, it depends what system we're talking about, because they have one system where they're putting a hollow needle in the skin and pumping drug through the hollow needle. So I think that's very close to a sub-Q injection.
All right, well, thank you. Let's thank Frank again.